Welcome back. So, in today's lecture, we will look at receptors. We will focus on understanding mainly the structure and how structure of receptors and how they function. Okay. So, receptors are, as we discussed earlier, are very important proteins which are by far the best or the most important targets in medicinal chemistry, and a number of diseases that we suffer from are related to receptors. So, for example, pain, depression, in some cases heart failure, asthma, and some certain neurodegenerative disorders are all in some way, shape or form connected to the dysfunction in receptors. So, to understand this, one needs to understand how communication system works between cells, especially in a complex organism. So, if for example, there are individual heart cells and each of these heart cells were to contract, let us say at different times, okay. So, then it would really lead to a chaotic system. So, one needs to make sure that the communication within and among these heart cells occurs in a very efficient manner, so that all the heart cells contract at the same time, okay, so that the blood can be circulated efficiently. Okay. So, using this similar concept, it is true for one can extrapolate these to other organs as well. So, all the organs have a coordinated function and these need to be controlled, and receptors are the ones which are responsible for this kind of coordination and control. Okay. So, and a lot of the coordination and communication system originates in the brain. Okay. So, here is a schematic of the brain, and which is what we call as the central nervous system, and of course, there is a spinal cord, and all of these contain neurons. Okay. So, these are the ones that generate the signal and transmit it. Okay. The signal that we are talking about is nothing but an electric pulse. So, this electric pulse travels down the nerve, which is also called the neuron, towards the target, right. And so, it communicates to the target whether, let us say, a muscle should contract or not. From the brain, a lot of these signals transmit and they control the functioning of the heart, the smooth muscles, the pupils for example, and then you also have the signal controlling how certain glands work, and so does for example, the adrenal glands or the sweat glands, and of course, including the excretory system is controlled by these types of signals. Okay. Now, if of course, if it is all that there was to signaling, it would be difficult to imagine how these drugs can actually affect this communication system. So, what is an important feature that is crucial to the understanding of how drugs work on receptors is that these systems are not directly connected. Okay. So, for example, neurons do not connect directly to their target cells. There is just a small distance, typically of maybe 100 angstrom, wherein there is a gap. Okay. So, this distance, this gap has to be covered, which the electrical pulse is unable to jump. So, what happens is that the signal gets all the way here and then it stops. So, the way in which this is further transmitted is through small chemical messengers. These are known as neurotransmitters, and these originate from the nerve cell. Okay. So, what happens is that the messenger travels that distance from the neuron to the target, and there is and across this gap, and then interacts with the specific protein or receptor that is on the surface, and then there is a signal that is given out. Okay. So, these chemical messengers, which are known as neurotransmitters, are primarily responsible for the signal that is transmitted. Okay. Now, what happens is that after the binding of the messenger to the target occurs, it results in a cascade of secondary effects. Okay. So, what these effects are could be, for example, flow of ions, which result in changing of the potential inside a cell, or it could be switching on or off an enzyme inside the target cell. So, for example, the signal of can be like contraction of a muscle cell, or it can also be metabolism, which in terms of stress where fatty acid metabolism is signaled in a fat cell. Okay. So, this signal that is being transmitted occurs within the cell. So, to recap, the neuron has an electrical impulse which is then transmitted up to a particular point, after which a messenger, which is usually a small molecule, carries that signal forward to the site of interest, wherein a receptor interacts with this messenger to transmit a signal. Okay. The first person to propose the existence of receptors was Langley. Okay. What he did was he conducted an experiment wherein he was able to show that certain target cells responded to a chemical neurotransmitter, which is nicotine, even when the neurons that were supplying these cells were dead. So, what it means is that he could figure out, or he demonstrated that it is a chemical messenger which interacts with the target. Second thing is that he also showed that there is a target which responds to chemical messengers. Okay. So, these proteins are, he coined the term as receptive substances, which are now called as receptors, and they respond to these chemical signals, which are now broadly two classes, which is hormones or neurotransmitters. Okay. So, when there is a chemical process that is involved, it is logical for us to propose that other chemicals such as drugs can interfere or interact with this process, and therefore, receptors can be good targets for drug discovery. So, if you look at the structures of some of the neurotransmitters, you will find that many of them are simple molecules such as monoamines. An example is acetylcholine, whose structure is shown here. It is a very simple small molecule. It has an amine which is positively charged. Okay. There are other examples such as dopamine or noradrenaline, and even a simple amino acid called as glycine or gamma aminobutyric acid, serotonin and glutamic acid. So, these are some examples of neurotransmitters which help in transmitting a signal from a neuron to the target site. You can also have fairly complex structures such as lipids, examples are prostaglandins, or purines such as adenosine or ATP, and sometimes you can also have neuropeptides such as endorphins, which can transmit these signals. Other examples are also peptide based hormones such as angiotensin or bradykinin, and even sometimes enzymes can do this, play this role of transmitting signals. Okay. In general, a neuron releases mainly one type of neurotransmitter, okay, and the receptor which is present on the target cell waits or responds for this signal to be to arrive, okay, or this neurotransmitter to arrive. Okay. So, the target cell is specific, or there is specificity in the target cell for the messenger. Okay. However, this does not mean that the target cell has only one type of receptor. So, in this schematic, so here is a target cell and here is one of the neurons that is generating this neurotransmitter, which I am showing in blue, to the receptor which I am again showing in blue. But this is not the only receptor that is present in the cell. You also have couple of other receptors that I have shown here. Each target cell has a large number of neurons communicating with it, and of course, they do not all use the same type of neurotransmitter, and each cell has a large number of receptors which can communicate with different types of neurotransmitters. Okay. So, the target cell will have other receptors for other neurotransmitters. For example, here you have this blue one which is interacting with neuron 1, and you may also have a red one which is interacting with neuron 2, and the third one interacts with neuron 3. Okay. Now, but there may be longer distances that the signal has to travel for the same for it to interact with the receptor. Okay. So, here for example, in neuron 2 the distance that is travelled is much longer when compared to neuron 1. Okay. Now, there are hormones which are also released into the circulatory system by glands. The example that we looked at was adrenaline gland, which releases adrenaline. Okay. So, this is a signal, adrenaline is a signal that when danger is anticipated. Okay. So, there is this fright, flight or fight kind of response that is given out. Okay. So, when the adrenal medulla gland releases adrenaline, what happens is the body is preparing itself for vigorous exercise. So, a gland can release this hormone, which then can go and interact with its receptor site. Okay. So, hormones and neurotransmitters can be distinguished by the route that they travel. So, in the case of a neurotransmitter, the neuron, which is here, communicates with the target, which is fairly close to it, whereas a hormone can be released into the bloodstream, and once the hormone gets into the bloodstream, it can interact with its target in various locations. Okay. The receptor on the cell interacts with both neurotransmitters and hormones, can interact with the receptor, and once the message is received, the cell responds and the internal chemistry adjusts itself so that a biological response happens. Okay. So, in this schematic you see that the blood supply through the blood supply a hormone can be produced, or into the blood supply a hormone can be released, and this hormone can interact with this receptor, and of course, you can have couple of neurons interacting with the target to give you a desired response. Receptors are typically identified by the specific neurotransmitter or hormone that interacts with them. Okay. So, for example, dopaminergic receptor is activated by dopamine, and cholinergic receptors are activated by acetylcholine. You also have hormones which are, for example, adrenaline, which interact with adrenoreceptors or adrenergic receptors. Okay. So, in the schematic below, we see that a dopaminergic receptor interacts specifically with dopamine, and acetylcholine interacts specifically with cholinergic receptors. Okay. Now, not all receptors are activated by the same chemical messenger in exactly the same way throughout the body. So, the example that we can look at here is between a heart and a lung, which has the same receptor, but they can be structurally slightly different, and the binding site is perhaps a little bit different. And so, what happens is that even though the same small molecule, in this case acetylcholine, interacts with the receptor in the heart, there could be different responses in the heart as well as in the lung. Okay. Now, these differences can be exploited to achieve what is known as selectivity. Let us say we want to selectively target the lung receptor over the heart receptor, then one could make structural variations in the molecule that we want, so that it specifically hits the lung and does not hit the heart. Now, in general, it is true that there are various types of a particular receptor, just like we saw heart and lung can be different, and these are normally identified by numbers or letters. Okay. Earlier, many of the receptors that were discovered were named after natural products. So, for example, muscarinic receptor or nicotinic receptors are the ones that are a subset of cholinergic receptors, which these respond to muscarine or nicotine. Okay. Now, as I mentioned earlier, the current emphasis is to design and develop selective receptor binding compounds. Okay. And not just receptor types, but they also the subtype has to be appropriate. What this will result in is much fewer side effects if you can achieve selectivity. So, a receptor is a protein which is usually located or embedded within the cell membrane. Now, since this is located in the cell membrane, it can interact with molecules which are outside the cell. Okay. The surface of the protein is quite complicated, and it can have hollows, ravines and ridges, and which form the right shape for the messenger to come and bind. Broadly speaking, in this area where the receptor where the messenger binds is known as a binding site. The analogy that we have learned before is the active site of an enzyme. So, the binding site of a receptor is very similar to the active site of an enzyme. Now, after the messenger binds, what happens is that there is a sort of a induced fit that occurs, and this switches on the receptor and the message is transmitted. Once the message is transmitted and the job is done, then the messenger can be released out from the receptor. Of course, the first thing that we need to note is that the messenger fits the binding site of a protein receptor perfectly, okay? And this fit what we call as an induced fit. So, induced fit is nothing but when you have the receptor sort of changing its shape, as we saw in a enzyme active site, to a little bit to bind the messenger. The important difference between enzymes and receptors is that the chemical messenger that transmits the signal in the case of a receptor remains unchanged, whereas in an enzyme the substrate which binds to the protein undergoes a chemical reaction and is transformed to another product. So, that is a very important difference between enzymes and receptors. Now, when it passes, once it fits into the binding site of the receptor and passes on the message, and if it is unchanged, then what exactly is happening? The question is, how does the chemical messenger tell the receptor its message, and how is this message conveyed to the cell? In order to understand this, let us imagine a hypothetical receptor which has a hydrogen bonding region, a van der Waals binding region and an ionic binding region. So, the hydrogen bonding is given by a hydroxyl group, and perhaps there is a region, a small aromatic residue which can result in van der Waals binding, and there could be a free carboxylate ion which does ionic binding. Now, let us imagine the ligand which binds to this receptor as the structure shown here. So, this structure has a aromatic ring which can do van der Waals binding. It also has a free hydroxyl group which can perhaps do hydrogen bonding to the hydroxyl group in the receptor surface, and it also has a ammonium salt which can bind to the carboxylate ion. Okay. So, it appears that the neurotransmitter and the receptor are very well placed to bind to each other. Now, after it binds, imagine that it binds, and if this interaction occurs, the van der Waals interaction occurs and the hydrogen bonding interaction occurs, and these are extremely complementary, and therefore, one would imagine that this would be a fairly good binding effect. But if you notice here, the ammonium salt is interacting with the carboxylate, but it appears that the fit is not perfect. Okay. So, therefore, the ionic interaction is not as strong as it could be. But if the receptor changes its conformation a bit, if it gets closer to the ammonium salt, then it is possible that this binding interaction becomes stronger, and that may be the right or optimum interaction which results in a strong binding. Okay. So, this model that we have looked at, it is a hypothetical model, but it helps us understand the concept of an induced fit. So, there are couple of interactions which are already quite strong, but maybe there are few other interactions that will happen if the protein can undergo a conformation change. And together, once this conformation change occurs or the series of conformation changes occur, then a strong interaction results. Okay. So, this is called as an induced fit. In reality, both the messenger and the binding site would probably undergo conformational changes to maximize the bonding forces between them.